Genetic engineering is a field of biotechnology that involves the manipulation of an organism's genetic material to achieve desired traits or characteristics. It encompasses a range of techniques and technologies used to alter the DNA, deoxyribonucleic acid, of organisms, including bacteria, plants, animals, and even humans. The primary goal of genetic engineering is to introduce specific genetic changes that would not occur naturally through mating or evolution. Here's a detailed overview of the genetic engineering process. Identification of desired trait. The first step in genetic engineering is to identify the specific trait or characteristic that researchers want to introduce or modify in an organism. This could involve traits such as increased crop yield, disease resistance, improved nutritional content, or enhanced medicinal properties. Isolation of genetic material. Once the desired trait is identified, scientists isolate the gene responsible for that trait from the organism's DNA. This can be done using various methods, such as PCR, polymerase chain reaction, or gene synthesis. Gene cloning. The isolated gene is then cloned using molecular biology techniques. Cloning involves inserting the isolated gene into a carrier molecule called a vector, often a plasmid, a small DNA molecule found in bacteria. The vector acts as a vehicle to transport the gene into the target organism cells. Recombinant DNA technology. The vector containing the cloned gene is introduced into the cells of the target organism. This can be achieved using methods such as transformation, for bacteria, viral vectors, for animals and plants, or gene guns, for plants. Integration and expression. Once the recombinant DNA is inside the target organism cells, the cloned gene integrates into the host genome. The host organism then begins to express the protein encoded by the cloned gene, resulting in the manifestation of the desired trait. Selection and screening. In many cases, not all cells will successfully incorporate the recombinant DNA. Therefore, researchers use selection markers, such as antibiotic resistance genes, to identify and isolate cells that have taken up the foreign DNA. Additionally, screening techniques are employed to confirm the presence and expression of the desired trait. Propagation and breeding. If the genetically engineered organism is a plant, researchers can propagate and breed it to create a population with the desired trait. This involves selecting individuals that exhibit the desired characteristic and crossbreeding them to stabilize and enhance the trait in subsequent generations. For animals, a similar process can be applied, although it might involve more complex breeding strategies. Ethical and safety considerations. Throughout the genetic engineering process, ethical and safety considerations are essential. Researchers must consider the potential risks and benefits associated with releasing genetically modified organisms into the environment or using them for various purposes, such as food production or medicine. It's important to note that genetic engineering has a wide range of applications beyond just creating genetically modified organisms. It has revolutionized fields such as medicine, gene therapy, agriculture, genetically modified crops, and industry, bioremediation and enzyme production, among others. However, ethical and regulatory discussions continue to surround the use of genetic engineering, particularly when it comes to its impact on ecosystems, biodiversity, and human health.